In this presentation, we are going to continue with notions in experimentation as an introduction into physiological reactions in advanced level biology code 0710 at the Cameroon GCE. My name is Lanyu Jude Fonbum. I am a holder of a Bachelor of Education degree in Life and Earth Sciences from the University of Marua, Cameroon, and a Doctor of Philosophy degree in Clinical Biochemistry from the University of Dishang, Cameroon. We are going to continue with the presentation of results of a scientific study. A scientific study can provide diverse information that need to be presented in a report. Results of descriptive studies, that is, the biological observation of nature, can be presented using symbols, diagrams, observation charts, nature's calendar, among others. However, we are going to dwell only on biological diagrams, understanding of which is crucial to succeeding with your practical work. In hypothesis-based studies based on experimentation, the variations of the dependent variable with respect to the independent variable are recorded as experimental data. These results can be presented in tables or graphs. The structure of a living thing or its part is best represented using a diagram. A diagram is a simple outline of an organism, object, or idea. It is made using lines, arrows, or shapes. To draw a diagram, we need a white and plain sheet of paper in order to have a good visibility. We need a soft HB pencil which gives clear dark lines. An eraser is also needed to correct the diagram when necessary. We also need a ruler for labeling. There are a set of rules to follow when drawing a biological diagram. A biological diagram is done with a free hand, no matter how perfect the part resembles a shape. Do not use, for example, a ruler to draw an apparently straight part or a compass for circular parts. Use neat lines. To achieve this, do not hang on your pencil, do not print too hard or too light. Before starting to draw a section of the diagram, have in mind your starting and your stopping points and do not stop along the way. Centralize your diagram and make it large. It should occupy between 50 to 75% of the drawing space provided while leaving space for labeling and annotation. Maintain proportions of parts of the structure. Small parts should be proportionately small and same for large parts. At the end, you must add a title to your diagram. After drawing a diagram, you add meaning to it through labeling and annotation. To label a diagram, we also follow a set of rules. Labeling lines should be straight, drawn using a ruler, and not the free hand. A labeling line should not have an arrowhead. Labeling lines should point and touch the correct part. Labeling lines should not cross each other. Labels should not be written on the diagram, no matter how large the part is. The title should be written under the diagram and must be underlined. You may also need to include the body section in which the diagram is drawn, that is, whole, longitudinal section, or transverse section. In other cases, you may instead add the body orientation used, for example, ventral view, dorsal, side, etc. Annotations give information about the parts labeled. An annotation is done on the diagram in brackets after the label of the part. It has two parts, a brief description of the part and the function, the two separated by a comma. Before delving into the presentation of experimental data, let us first briefly look into the notion of data and experimentation. Data is factual information, often in the form of worded facts or numerical figures, obtained from experiments or surveys, used as a basis for making calculations or drawing conclusions. Scientists collect two types of data, qualitative data and quantitative data. Qualitative data is description in words of what is observed. They are based on some quality of an observation, such as color, odor, or texture. This type of data has a limitation in that it is subjective. That is, it is based on the appreciation of the observer. Quantitative data, on the other hand, is numeric measurements. They include measurements using specialized equipment such as mass, volume, temperature, distance, concentration, time, or frequency. The data are objective. That is, it's the same no matter who does the measures because they are done using equipment. Tables are a fundamental tool for organizing and presenting data in a clear and concise manner. However, they often fail to bring out the relationship between variables. The following are some simple types of tables that can be useful in presenting reports of physiological experiments or projects. A simple table or a one-way table presents data for a single characteristic or variable. In the example, we see the variable is the body mass index status and the frequency of individuals in this group. A frequency distribution table groups data into classes or intervals and shows the frequency of each class. In the example, we suppose that in your project, you have gone to a local health facility and have taken from the records information about malaria and have categorized the cases into age groups. A contingency table or two-way table shows the relationship between two or more variables. The example is data from a study seeking the relation between use of mosquito nets as protection against malaria and development of the disease. We suppose that the students sampled two groups of people, one of those who use mosquito nets and the other who do not, and then test them for the presence of plasmodium in their blood. A data table presents raw data in a structured format, often used in spreadsheets, for example, Microsoft Excel or databases. In the example, we have a project to evaluate the health status of students in a given class. The students collect the various vital parameters that will serve in identifying health conditions, for example, obesity and hypertension. Let us now look at the presentation of data using graphs. A graph is a diagram used to indicate relationship between two or more variable quantities. The quantities are usually measured along two axes set at right angles to each other. Graphs come in different forms. Let us take the first type of graph, which is a simple line graph. A line graph represents the cause and effect relation between two or more variables plotted in intersecting straight lines with two axes, the x and the y. The independent variable is plotted on the x-axis and the dependent variable on the y-axis. Let us take as example, representing the enzyme concentration against the initial speed of the reaction. The enzyme concentrations are set by the researcher. So enzyme concentration is the independent variable and is put in the x-axis. On the other hand, the initial rate of the reaction is measured during the experiment, and thus it is the dependent variable plotted on the y-axis. A bar chart is a graph that represents categorical data with rectangular bars with heights proportional to the values they represent. The bars represent discrete categories being measured. In the example, the type of tissue is the discrete category, that is the bars, while the catalase activity of the tissue is depicted by the height of each bar. Another example is the number of births in various age groups. A histogram is a statistical graph of a frequency distribution in which vertical rectangles of different heights are proportionate to corresponding frequencies. It differs from the bar chart in that it studies the continuous variation of a single variable over a given range of values called the bin. In the example, we see the numbers of births according to the months of the year. Here, the month does not influence the number of births, unlike in the example of the bar chart, where the type of tissue influences the catalase activity or the age group influences the number of births. A pie chart is a circular graph which is divided into sectors to illustrate numerical proportions. It used to represent data that sum up to a whole, for example, 100%. The arc length, area, or central angle of each sector is proportional to the quantity it represents. Suppose in a project, you want to present the body mass indices of a class with students distributed as 8 underweight, 58 normal weight, 32 overweight, and 14 obese. To plot a pie chart, first sum the values of all the components, values. Summing the subjects in this study gives 112 students. We then use the sum to calculate each proportion. For example, the students with normal weight will constitute 52% of the students. Next, we convert the proportion to an angle in a circle, knowing that the sum of central angles of a circle is 360 degrees. For example, since the normal weight students constitute 52% of all the students, the normal weight status will thus take 52% of 360, which gives 187 degrees. We do same for the other values and have their angles. After getting all the angles, we use a compass to construct a circle and then a protractor to mark out the various angles. We have come to the end of this presentation on the process of scientific inquiry. In the next presentations, we will be taking the various physiological reactions. Thanks for your time. Hope you enjoyed following up the presentation. Please subscribe to this channel, like, and share.